HeLa Cells. In 1951, a woman named Henrietta Lacks entered Johns Hopkins Hospital in Baltimore seeking treatment for an aggressive form of cervical cancer. At the time, Hopkins was one of the few hospitals that treated Black patients, and the procedures she underwent followed the medical standards of the era. During her treatment, physicians removed small samples of her tumor tissue. This was routine practice. Consent was not requested. It was not considered necessary. What happened next would permanently alter the course of modern medicine. When Henrietta's cells were placed into laboratory culture, researchers noticed something unprecedented. Most human cells die after a limited number of divisions. They weaken, degrade, and stop replicating. Henrietta's cells did not. They divided rapidly and continuously, showing none of the normal signs of cellular aging. They appeared biologically unstoppable. For the first time, scientists were observing a human cell line that could survive indefinitely outside the body. The cells were labeled HeLa, derived from the first two letters of Henrietta Lacks' first and last names, deliberately obscuring her identity. These cells spread quickly through research institutions. HeLa cells proved remarkably durable, easy to grow, and extraordinarily consistent. They became essential to laboratory science. Researchers used them to develop the polio vaccine, to study how cancer cells behave, to map genes, and to understand how viruses invade human tissue. They were exposed to radiation, toxins, and extreme temperatures. They were sent into space to examine how human cells respond to microgravity. Entire disciplines, virology, genetics, immunology, and cell biology, advanced decades faster because of HeLa cells. Yet the woman behind the cells remained invisible. Henrietta Lacks died later that same year, unaware that parts of her body were still alive and multiplying in laboratories around the world. Her family was not informed. Her name was omitted from publications. Meanwhile, HeLa cells were mass-produced, distributed globally, and used in tens of thousands of studies. They became one of the most widely utilized biological materials in history, generating enormous scientific and commercial value. It was not until the 1970s that Henrietta Lacks' family learned the truth. Researchers contacted them seeking genetic information, not to explain the legacy of the cells, but to help distinguish HeLa DNA from contaminated samples and other experiments. The family was shocked to discover that Henrietta's cells had been living on for decades. This revelation ignited serious ethical debates about consent, patient rights, and ownership of biological material. At the time the cells were taken, no laws had been broken, but many questioned whether something deeply human had been ignored. Today, Henrietta Lacks is recognized as a foundational figure in medical science. Her cells are still used in cutting-edge research, including cancer therapies and studies related to COVID-19. The scientific value of HeLa cells is undisputed, but their story stands as a reminder that progress often advances faster than ethics, and that one of the most important discoveries in medicine came from a woman who was never asked, never informed, and nearly erased from the record. Hand hygiene truth. In the mid-19th century, hospitals were places of learning, prestige, and authority. They were also places where women routinely died after childbirth at rates so high they were considered unavoidable. In Vienna's General Hospital, one maternity ward had a mortality rate from childbed fever several times higher than the other. This discrepancy was well known, carefully recorded, and largely ignored. It was within this environment that a Hungarian physician named Ignaz Semmelweis made an observation that would quietly challenge the foundations of modern medicine. Semmelweis noticed a disturbing pattern. The ward staffed by doctors and medical students consistently had far more deaths than the ward run by midwives. The difference could not be explained by overcrowding, climate, or patient demographics. Women themselves recognized the danger. Many begged not to be admitted to the doctor's ward. Some gave birth in the street to avoid it. Semmelweis did what few of his contemporaries bothered to do. He compared the data without assuming the outcome. The breakthrough came after the death of a colleague who had been cut during an autopsy and later developed symptoms identical to childbed fever. Semmelweis concluded that something was being carried from cadavers to living patients. Doctors routinely moved directly from dissection rooms to delivery beds, their hands visibly soiled. At the time, there was no germ theory. Disease was blamed on imbalances, miasma, or fate. Still, Semmelweis acted. He ordered all staff to wash their hands with a chlorinated lime solution before examining patients. The results were immediate and undeniable. Mortality rates collapsed, dropping from catastrophic levels to near zero in some months. The numbers were precise, repeatable, and documented. Semmelweis had found a way to save lives using nothing more than soap, water, and discipline. Yet instead of acceptance, his findings were met with hostility. Many senior physicians rejected the idea that they themselves were responsible for the deaths. The implication was deeply offensive. It suggested that respected doctors, symbols of healing, were agents of disease. Semmelweis struggled to communicate his conclusions within the rigid academic culture of the time. He published poorly, argued emotionally, and lacked a theoretical framework to explain his results. Without germ theory, his evidence was dismissed as coincidence or exaggeration. Over time, opposition hardened. Semmelweis was ridiculed, isolated, and eventually removed from his hospital position. His ideas were not debated so much as ignored. The handwashing protocols were abandoned. Mortality rates rose again. Thousands of deaths followed that could have been prevented. Semmelweis, increasingly distressed, continued to speak out. His behavior grew erratic. In 1865, he was committed to an asylum under disputed circumstances. He died shortly after, reportedly from injuries sustained during his confinement. The cause of death was infection. Ironically, it was the very type of condition his work had sought to prevent. Only years later, after the acceptance of germ theory, were his findings recognized as correct. By then, the damage was irreversible. Today, hand hygiene is one of the most fundamental principles in medicine, but its adoption came at a terrible cost. The data existed. The solution worked. What failed was the willingness of an institution to accept evidence that threatened its authority. Semmelweis did not disappear because he was wrong. He disappeared because he was inconvenient. Inheritance laws. In the mid-19th century, inside a quiet monastery in what is now the Czech Republic, a man was patiently counting pea plants. There were no surgical theaters, no laboratories filled with glassware, no grand institutions watching his work. Gregor Mendel was an Augustinian monk with formal training in mathematics and natural science, and his experiments were carried out in a garden. What he uncovered there would eventually become the foundation of modern genetics. At the time, almost no one noticed. Between 1856 and 1863, Mendel conducted thousands of controlled crossbreeding experiments using pea plants. He chose them carefully. Peas had clear, discrete traits: round or wrinkled seeds, yellow or green color, tall or short stems. Mendel tracked these traits across generations with meticulous precision. Instead of blending together, the traits behaved in predictable patterns. Some appeared, others vanished, only to re-emerge later. Mendel realized that inheritance followed mathematical rules. In 1865, he presented his findings to the Natural History Society of Brno. He described what we now call dominant and recessive traits, the law of segregation, and the idea that hereditary factors are passed down as distinct units. These conclusions directly challenged prevailing beliefs. At the time, most scientists believed inheritance was a vague blending process, not a system governed by countable elements. Mendel's work suggested otherwise. The following year, his paper was published under the title Experiments on Plant Hybridization. It was dense, statistical, and written in an unfamiliar language for biologists of the era. Mendel relied heavily on mathematics, a tool rarely applied to biology at the time. His results were precise, but they did not align with the dominant scientific debates of the century, which were focused on evolution, natural selection, and visible variation in populations. As a result, the paper was largely ignored. It was cited rarely and discussed even less. Mendel sent copies to prominent scientists across Europe. Few responded. Those who did failed to grasp its significance. The problem was not that Mendel was wrong. It was that his findings existed in isolation. There was no framework connecting his factors to cells, chromosomes, or evolution itself. Without that context, his work appeared narrow and irrelevant. Mendel eventually abandoned his experiments. Administrative duties consumed his time after he became abbot of the monastery. He died in 1884, unaware that he had identified the basic mechanism of heredity. For more than 3 decades, his paper remained buried in obscurity, quietly archived, its implications untouched. Then, around 1900, several scientists working independently on heredity encountered Mendel's forgotten work. They realized that his observations explained their own results with startling clarity. Almost overnight, Mendel's laws were rediscovered and validated. What had once seemed obscure became essential. Genetics emerged as a scientific discipline, built directly on principles Mendel had described decades earlier. The delay had consequences. An entire generation of biological research proceeded without an understanding of how traits are inherited. Evolutionary theory advanced without genetics. Medicine lacked insight into hereditary disease. The knowledge existed, written down and published, but it was not absorbed. Mendel's story is not one of suppression by force, but of neglect by consensus. His work was not hidden behind locked doors. It was overlooked because it did not fit the intellectual priorities of the time. In the history of science, few discoveries demonstrate more clearly how transformative knowledge can remain invisible when no one is prepared to see it. Zealandia Continent. For much of modern geology, continents were thought to be obvious things. Large, continuous land masses rising clearly above sea level, bordered by oceans and defined by clear edges. By that definition, the map of the world seemed complete. And yet, scattered across the southwestern Pacific were clues that did not fit. Islands such as New Zealand, New Caledonia, and a chain of submerged plateaus contained rocks that looked continental, not oceanic. For decades, these anomalies were noted, cataloged, and set aside. The problem was scale. Individually, these islands could be explained as fragments, microcontinents, or geological oddities. But collectively, they suggested something much larger. Geologists observed that the crust beneath them was thicker and less dense than typical ocean floor. The rocks were ancient, some hundreds of millions of years old, unlike the relatively young basaltic crust that forms most of the seabed. Fossils, mineral compositions, and structural patterns hinted at a shared history. Throughout the 20th century, the idea that these landforms were connected remained controversial. Mapping the ocean floor was difficult, expensive, and incomplete. Without clear visual evidence, the hypothesis of a submerged continent struggled to gain traction. Traditional geological models did not easily accommodate a continent that was almost entirely underwater. Continents, by convention, were expected to be visible. As technology improved, the picture began to change. Deep-sea drilling retrieved continental-type rocks from beneath thousands of meters of water. Satellite gravity measurements revealed a broad, coherent region with boundaries distinct from the surrounding oceanic crust. Seismic surveys showed a continuous block of stretched continental lithosphere extending across millions of square kilometers. Piece by piece, the data aligned. What emerged was the outline of a vast landmass now known as Zealandia. Covering nearly 5 million square kilometers, it is roughly 2/3 the size of Australia. More than 90% of it lies submerged. Only its highest elevations remain above sea level, forming the islands we recognize today. Geological evidence indicates that Zealandia was once more elevated. Over tens of millions of years, tectonic stretching thinned its crust, causing it to subside gradually beneath the Pacific. Acceptance was slow, not because the evidence was falsified or suppressed, but because it required a shift in how continents were defined. Many geologists questioned whether a mostly submerged landmass qualified. Others argued that it blurred the distinction between continents and continental fragments. The debate continued as new data accumulated. In 2017, a comprehensive analysis synthesized decades of research. The Geological Society of America formally recognized Zealandia as Earth's 8th continent, based on crustal thickness, geology, elevation relative to the surrounding ocean floor, and clearly defined boundaries. What had once been treated as disconnected anomalies was finally understood as a single, coherent geological entity. Even now, much of Zealandia remains unexplored. Large areas have never been directly sampled. Scientists continue to investigate when and how it submerged, what ecosystems may have existed when more of it was above water, and how its breakup influenced the Pacific region. The discovery of Zealandia stands as a reminder that entire chapters of Earth's history can remain hidden, not because they are deliberately concealed, but because they lie just beyond the limits of what we expect to find. Water memory debate. In 1988, one of the world's most respected scientific journals published a paper that immediately unsettled the foundations of chemistry and biology. The study, led by French immunologist Jacques Benveniste, claimed that water could retain a memory of substances once dissolved in it, even after those substances had been diluted to the point where no molecules remained. If true, the finding contradicted basic principles of molecular science and appeared to lend credibility to homeopathy, a practice widely dismissed by mainstream medicine. Benveniste's team reported that human basophils, a type of white blood cell involved in immune responses, reacted to solutions that should have been chemically inert. According to the paper, water that had once contained antibodies could still trigger a biological response after extreme serial dilution. The implication was extraordinary. Water itself might preserve structural information independent of the molecules originally present. Nature published the paper, but not without hesitation. Alongside it, the editors added an unusual disclaimer, stating that the claims were so implausible that they required extraordinary scrutiny. This was not a routine peer-reviewed acceptance. It was a warning. The journal made clear that the results challenged established physical laws and demanded independent verification. What followed was unprecedented. Nature organized an on-site investigation of Benveniste's laboratory. The review team included the journal's editor John Maddox, chemist Walter Stewart, and magician and skeptic James Randi, whose role was to identify potential sources of unconscious bias or experimental error. Under controlled, blinded conditions, the reported effects disappeared. When the researchers no longer knew which samples were which, the results no longer held. Nature later published a follow-up report concluding that the original findings could not be reliably reproduced and were likely the result of methodological flaws. Replication attempts by other laboratories around the world also failed. The concept of water memory was rejected by the broader scientific community, which pointed out that liquid water constantly rearranges its hydrogen-bonded structure on timescales of trillionths of a second, making long-term molecular memory incompatible with known physics. The controversy did not end there. Benveniste maintained that his work had been unfairly targeted and that institutional skepticism had crossed into suppression. He continued to explore related ideas, later proposing that biological signals could be digitally recorded and transmitted, claims that were again met with rejection. His funding declined, his reputation suffered, and his work became a cautionary tale within mainstream science. Critics argue that the case demonstrates science functioning as it should, extraordinary claims tested, challenged, and discarded when they fail replication. Supporters counter that the reaction revealed how aggressively the scientific establishment defends its boundaries, especially when results threaten entrenched assumptions. What is undeniable is that the episode left a lasting mark. Nature's public intervention was rare, and the debate surrounding the paper remains one of the most discussed editorial decisions in modern scientific history. The Benveniste affair is remembered not because water memory was accepted, but because it exposed how science responds when data collide with theory. Whether seen as a necessary correction or a warning about institutional power, the controversy continues to be cited as an example of how certain ideas are not merely disproven, but decisively pushed to the margins. Plastic eaters. In 2016, researchers investigating waste at a plastic recycling facility in Japan encountered something they were not looking for. Amid piles of discarded bottles and synthetic debris, They identified a microorganism growing directly on the surface of polyethylene terephthalate, or PET plastic. This material was designed to resist degradation. It was considered durable, stable, and largely immune to natural breakdown. Yet the bacterium was not merely present. It was feeding. The organism was later named Ignaz Semmelweis. What made it remarkable was not its existence, but its behavior. Laboratory analysis showed that it could use PET as a primary energy source. Instead of plastic persisting for centuries, this microbe was slowly dismantling it, molecule by molecule, as part of its normal metabolism. The process was not fast. It was not dramatic, but it was real. Further study revealed how it worked. Ignaz Semmelweis produces two enzymes, now known as PETase and MHETase. PETase initiates the breakdown by attacking the long polymer chains that give plastic its strength. This reaction reduces PET into an intermediate compound. MHETase then completes the process, converting the fragments into simpler molecules, including terephthalic acid and ethylene glycol. These are substances the bacterium can absorb and use for growth. From a biochemical standpoint, the finding was unexpected. PET had only been in widespread use since the mid-20th century. Its resistance to natural degradation was one of its defining features. The existence of an organism capable of exploiting it suggested that microbial evolution had already begun adapting to human-made materials. Nature, it seemed, had started responding to a problem humanity had created. The discovery did not immediately dominate headlines. Researchers approached the findings cautiously. The degradation rate was slow under natural conditions, and the bacterium functioned optimally only within a narrow range of temperatures. There was no suggestion that it could solve the global plastic crisis on its own. Instead, the initial response focused on verification, enzyme structure, and understanding whether the process could be enhanced or replicated safely. Within scientific circles, the implications were clear. The enzymes offered a potential blueprint. If their efficiency could be improved through engineering, they might one day be used to accelerate plastic recycling or reduce environmental accumulation. Subsequent studies explored modifying PETase to increase its activity, while others examined whether similar organisms existed elsewhere. Public awareness came later, often stripped of nuance. Headlines emphasized plastic-eating bacteria, implying rapid cleanup and simple solutions. In reality, the discovery represented something subtler. It showed that plastic is not as biologically inert as once believed. It also demonstrated that ecosystems are already being altered at the microscopic level by synthetic materials. The significance of Ignaz Semmelweis lies less in what it can currently do, and more in what it reveals. Plastic had entered the environment. Life responded. The process was quiet, slow, and easily overlooked. But it marked a shift. For the first time, a material engineered to outlast nature had become part of a natural food chain, not by design, but by adaptation. Microplastic plague. In the 1990s, marine scientists began noticing an unusual phenomenon in their routine ocean surveys. Tiny specks appeared inside the bodies of fish, plankton, and other microscopic sea creatures. Initially, these particles were dismissed as sand, natural detritus, or harmless sediment drifting in the currents. But closer examination under the microscope revealed a far more troubling reality. The specks were fragments of plastic, often smaller than a millimeter, yet persistent and pervasive. The implications were staggering. These microplastics were not limited to the surface or shorelines. They had infiltrated the very foundation of marine ecosystems. Zooplankton ingested the particles, oblivious to their synthetic nature. Small fish consumed the plankton, and larger predators fed on those fish, allowing the fragments to accumulate and concentrate as they moved up the food chain. Over time, it became clear that humans were not exempt. Seafood on our plates, salt from the sea, and even drinking water carried traces of these microscopic plastics. What had once seemed like scattered pollution was, in fact, a global contamination issue unfolding silently across oceans, rivers, and lakes. Early research on microplastics spread cautiously through academic journals and conference presentations. Scientists were aware of the potential alarm the findings could cause. The particles were tiny, the data complex, and the scale of contamination still being assessed. There was no deliberate suppression, but the initial dissemination remained limited to specialized scientific circles. Meanwhile, industries producing and distributing plastic were slow to acknowledge or respond to the problem. What began as a subtle anomaly in marine samples would later emerge as a crisis affecting ecosystems and human health alike. As studies progressed, the scope of microplastic contamination widened. Microplastics were discovered in the deep sea, trapped in Arctic and Antarctic ice, embedded in river sediments, and even suspended in the air. These findings demonstrated that plastic debris does not remain localized. It travels, persists, and integrates into environments once thought pristine. Each new study confirmed that the problem was not temporary or surface level. It was structural, systemic, and global. The early researchers who first documented microplastics understood that they were observing the beginning of a slow-moving ecological catastrophe. The particles are resilient, chemically stable, and nearly impossible to remove once dispersed. The oceans, long considered a vast buffer against human activity, were proving to be far less forgiving than imagined. What had once seemed like a minor inconvenience, tiny bits of plastic drifting unnoticed, was now recognized as a foundational environmental issue that would take decades to fully quantify and mitigate. Today, microplastics are acknowledged as a ubiquitous pollutant, silently infiltrating food chains, water supplies, and natural habitats worldwide. Yet, the initial decades of study remain crucial to understanding how this problem emerged. Scientists who first sounded the alarm did so quietly, often ignored or met with skepticism. Yet their work laid the groundwork for one of the most pressing environmental challenges of the 21st century. The ocean's hidden plastic soup, once invisible to the naked eye, is now a stark reminder of how human activity reshapes even the most remote corners of the planet. Quark Model. In 1964, the world of particle physics was in turmoil. Protons and neutrons were understood as fundamental components of atomic nuclei, but the forces binding them and their internal structure remained mysterious. Amid this uncertainty, an American physicist named George Zweig proposed a radical idea. He suggested that protons, neutrons, and other hadrons were not indivisible, but composed of smaller constituents he called aces. These aces were a way to explain the observed patterns in particle behavior and properties, a systematic framework that could bring order to a rapidly growing catalog of subatomic particles. Zweig's proposal was groundbreaking. By suggesting that matter had a deeper layer, he challenged the prevailing notion of elementary particles. His aces were later recognized as what we now call quarks, a concept central to the standard model of particle physics. Yet, despite the brilliance of his idea, it failed to gain immediate traction. His initial paper, submitted for publication, was rejected. Peer reviewers were skeptical, not because the theory lacked mathematical consistency, but because it was too speculative, too revolutionary, and, in the climate of 1960s physics, too disruptive to established frameworks. Complicating the story was the parallel work of Murray Gell-Mann, a physicist working independently at Caltech. Gell-Mann proposed a remarkably similar model, coining the term quark, inspired by a line in James Joyce's Finnegan's Wake. His paper was accepted and widely publicized, overshadowing Zweig's contributions. Within months, the physics community largely credited Gell-Mann, while Zweig's aces remained largely unacknowledged. The distinction between the two was more than semantic. It was a matter of recognition, professional influence, and institutional politics. The delay in acknowledging Zweig's work was not the result of falsification or deliberate concealment, but of systemic dynamics within the scientific community. CERN and other research institutions were already engaged in intense rivalries and debates. Publication opportunities were scarce, and competing models were evaluated not only on scientific merit, but also on perceived authority and prestige. In this environment, Zweig's paper, despite its originality, slipped into obscurity. Years later, as experiments confirmed the existence of quarks through deep inelastic scattering and particle collision data, the importance of Zweig's contributions became undeniable. Retrospective acknowledgement restored his place in history, highlighting that multiple scientists had independently arrived at the same revolutionary insight. Today, the Quark model underpins particle physics, explaining not only the structure of matter, but also the interactions that govern atomic nuclei. Zweig's experience underscores the human dimension of scientific discovery. Ideas, no matter how correct or predictive, can be delayed, buried, or marginalized due to timing, institutional politics, and the social hierarchies of academia. His work, initially hidden in the shadow of a colleague, serves as a reminder that scientific progress is not always a straight path from insight to recognition, and that some breakthroughs require persistence, patience, and a willingness to wait decades for validation. Gene drives. In the early 2000s, a revolutionary idea emerged from the world of genetics, one that seemed more like science fiction than science fact. Researchers discovered a method to bias inheritance in living organisms, ensuring that a specific gene could spread through an entire population far faster than normal Mendelian genetics would allow. This technology became known as a gene drive. While conceptually simple, its implications were profound. It offered humanity a way to influence evolution itself, potentially reshaping ecosystems on a massive scale. One of the earliest and most compelling applications focused on mosquitoes. These insects are notorious carriers of deadly diseases like malaria, dengue, and Zika. By engineering a gene drive to disable the mosquito's ability to transmit the malaria parasite, scientists imagined a future where millions of lives could be saved each year. Laboratory studies demonstrated the promise almost immediately. In controlled populations, the engineered genes spread through successive generations at rates previously thought impossible, rapidly becoming fixed in the population. For the first time, it seemed feasible to alter a species intentionally, not just in small, localized ways, but at a scale that could affect entire ecosystems. Yet the very success of gene drives raised urgent and unprecedented concerns. Unlike conventional genetically modified organisms, these modifications cannot easily be recalled once released into the wild. The ecological consequences were difficult to predict. What would happen to predators that depended on mosquitoes? Could altering one species trigger cascading effects on pollination, food webs, or biodiversity? Could unforeseen mutations produce entirely new risks? These questions kept researchers cautious, tempering their excitement with a sense of profound responsibility. As a result, much of the early gene drive research occurred behind closed doors. Labs were highly controlled, experiments carefully monitored, and discussions often restricted to specialized policy meetings. Scientists, ethicists, and international organizations worked together to draft protocols, assess potential risks, and debate moral limits before releasing any findings to the wider public. The work was not hidden in the sense of secrecy or suppression, but the public conversation had to wait until researchers were confident enough to present their conclusions responsibly. The gene drive debate quickly expanded beyond laboratories. Governments and global organizations began considering frameworks for environmental release, weighing potential health benefits against the risks of irreversible ecological change. Public scrutiny, once the research was ready to be disclosed, highlighted the tension between innovation and caution. Even today, gene drives remain a subject of rigorous study, heated debate, and ethical deliberation. Few technologies in history have offered humans such direct control over the natural world. Gene drives present the tantalizing possibility of eradicating disease-carrying insects, yet they carry a weighty responsibility. Every step forward demands careful calculation, international oversight, and humility before the complexity of life itself. The discovery illustrates a broader truth. Scientific power often arrives before society is fully prepared to wield it, leaving researchers to navigate uncharted ethical and ecological territory. Lake Vostok microbes. Beneath 2 miles of ice in the heart of Antarctica lies a place no sunlight has touched for more than 15 million years. This is Lake Vostok, a massive subglacial lake, isolated from the surface, sealed under crushing pressure, and trapped in complete darkness. For decades, scientists knew of its existence through radar surveys and ice-penetrating sonar, but the lake remained entirely inaccessible. Its isolation created one of the most extreme environments on Earth, a place where life as we know it seemed impossible. In the late 20th and early 21st centuries, Russian researchers began drilling through the ice sheet to reach its waters. The task was painstaking. Any contamination from the surface could compromise the integrity of the samples, creating false positives for life. When the first water and sediment samples finally emerged from the depths, the results stunned the scientific community. Signs of microbial life were detected, genetic traces of bacteria and other microorganisms capable of surviving in total darkness, under immense pressure and without oxygen. These organisms had been sealed away for millions of years, evolving independently in conditions previously thought uninhabitable. The discovery raised profound questions. Life existed in isolation, entirely cut off from sunlight and conventional food sources, yet it had persisted. How had these microbes adapted to the extreme cold, the high pressure, and the lack of nutrients? Their existence suggested that life is far more resilient than imagined, capable of enduring in environments previously considered sterile. If life could survive in Lake Vostok, could it not also survive on icy moons such as Europa or Enceladus? The lake became a natural laboratory for astrobiology, hinting that life beyond Earth might not be as improbable as once assumed. Despite the potential significance, the results were not immediately shared with the public. The delay was not due to intentional concealment, but caution. Researchers had to verify that the samples were uncontaminated, reproduce their findings, and ensure that the evidence truly came from the lake itself rather than drilling equipment or the ice above. The high stakes of working in such an extreme environment required rigorous protocols and careful peer review. International collaboration added further complexity, as multiple nations coordinated to prevent contamination, share data, and confirm the findings independently. By the time the full story reached the wider world, Lake Vostok had already gained legendary status in scientific circles. It was no longer simply a body of water beneath ice. It represented a hidden frontier, a window into Earth's past, and a testbed for understanding life in extreme conditions. Microbes that thrived without sunlight, without oxygen, and isolated for millions of years reminded the world that the boundaries of life are far broader than once believed. Even today, Lake Vostok continues to intrigue researchers, a frozen world of mysteries waiting to be understood, proving that some of the most alien environments are not on distant planets. They exist right here on Earth. If you love uncovering the secrets they don't want you to know, hit subscribe and join Strange Wise, where the bizarre meets the truth.